I'm Dr. Michael Jain, and I'm an assistant member in the Department of Blood and Marrow Transplant and Cellular Immunotherapy at Moffitt Cancer Center. I'm pleased to discuss one of our recent works that are going to be presented at the SITC conference. This is a collaboration between Moffitt Cancer Center and a group at the University of Miami. And what we did in this study is we looked at the tumor biology, the lymphoma genomics, that associated with good or bad response after a new type of therapy called CAR T-cell therapy. In patients who receive CAR T-cell therapy, approximately 40% will go on to a long-term remission if they have diffuse large B-cell lymphoma. However, still 60% of patients will relapse. So we used a technology called whole genome sequencing to analyze the features of the tumor that may or may not associate with response after CAR T-cell therapy. And what we found is that the more complicated the set of genomic rearrangements that occurred in the tumor, the less likely they were to respond to CAR T-cell therapy. So for example, patients who had a certain type of chromosomal changes called chromotripsis, where we really see many different types of chromosomal changes, such as the chromosomes looking like they're blowing up, these patients had a lower response to CAR T-cell therapy. So now that we've identified these changes, the next step will be to try and figure out ways to target these types of patients and see if we can't improve their outcomes, either adding a drug to CAR T-cell therapy or to select these patients for other types of therapies. Now, to do this, we're going to have to integrate some of the biology, some things that we can find out by another technology called RNA sequencing, to really find out what are the immune cells in the immune environment that associate with these changes. But this is a really important first step to identifying the types of tumor biology that associate with good or poor response after CAR T-cell therapy with an eye to the future.